I will be talking about the association between heat shock protein 70 gene polymorphisms and outcomes in severe sepsis in Indian patients. I'd like to start with a question. Do our genes play a role in determining the outcome in sepsis? Now, sepsis, as we all know, is a common cause of ICU admission. The American Center for Disease Control calls it the second leading cause of death in non-coronary ICU patients and the 10th most common cause of death in their country. A study done in 2006 in 12 ICUs in Calcutta showed that severe sepsis accounted for 13% of all admissions and had a mortality of above 50%. Sepsis is characterized by excessive systemic inflammation in response to an infection. This is due to dysregulation and an imbalance between pro- and anti-inflammatory cytokines, resulting in excessive outpouring of pro-inflammatory cytokines and chemokines. The factors that determine the outcome in a patient with sepsis are host factors, pathogen factors, and therapy. The pathogen factors include the virulence of the organism and the size of the inoculum. There are numerous host factors such as nutritional status, coexisting illnesses, and genetic factors relating to the immune response to sepsis. And it is one of these genetic factors that we have studied. Heat shock protein 70 is a ubiquitous protein first discovered in the 1960s. These proteins are also known as stress proteins. It was discovered in the fruit fly when the fly was accidentally incubated at a higher temperature. Later, when they were studying the chromosomes, they realized that an unknown protein was produced. This led to the discovery that when cells are subjected to stress or shock, such as heat, an unknown protein was produced, which came to be known as heat shock protein. It has numerous functions, and it functions within the cell as a chaperone, assisting in protein folding, unfolding, translocation, degradation, and signal transduction. It's present extracellularly as well, and here it helps activate the innate immune system. It processes and presents immunogenic peptides to cytotoxic T cells, resulting in a pro-inflammatory cytokine secretion. Now, sepsis, as we all know, is characterized by endotoxemia. The endotoxin lipopolysaccharide of the pathogen interacts with host immune cells and activates them through multiple pathways, the most common and best described of which is the toll-like receptor or TLR4 pathway. In a normal immune response, the pathogen is eliminated. However, if there are abnormalities of the TLR4 pathway, this results in an abnormal or dysregulated immune response, resulting in the excessive outpouring of cytokines. And it is well established that abnormalities in the TLR4 pathway affect sepsis. As HSP70 is also known to be an alternate pathway for immune activation, it is likely that abnormalities in this pathway will also affect sepsis. Polymorphisms, in particular single nucleotide polymorphisms, or SNPs, are common in the HSP70 gene. Certain SNPs, for example, the HSP-A1B polymorphism and the HSP-A1L polymorphism, have been shown in patients with severe multiple trauma to be associated with higher blood levels of TNF-alpha and interleukin-6. It is also associated with an increased risk of liver failure and higher organ failure scores. The HSP-A1B polymorphism has also been shown to be associated with an increased risk of septic shock in adult patients admitted with community-acquired pneumonia. HSP-A1267G, RS18438, and RS1043618 polymorphisms have also been shown to be associated with an increased susceptibility to high-altitude pulmonary edema in Chinese railway construction workers. The latter two polymorphisms, RS18438 and RS1043618, have been shown to be associated with an increased susceptibility to coronary artery disease in the Chinese population. We therefore hypothesized that specific genetic polymorphisms in the HSP70 gene may determine the outcome in patients admitted to the ICU with a diagnosis of sepsis. The 4 polymorphisms we looked at were HSP-A1267G, HSP-A1L, RS1008438, and RS1043618. This was a prospective cross-sectional study carried out in the medical ICU of the Christian Medical College, Vellore, between August 2009 and February 2010, after being approved by the Institutional Review Board. All patients with the diagnosis of sepsis were recruited. Exclusion criteria included hematological malignancy, febrile neutropenia, HIV infection, immunosuppression, and failure to get consent. Consecutive patients meeting the inclusion criteria were enrolled. Written informed consent was obtained from the next of kin. A 15 ml sample of venous blood was taken, and clinical data was recorded in a predetermined format. The primary outcome of interest was acute organ failure, and the organ systems we looked at were the cardiovascular, CNS, respiratory, renal, hematologic, metabolic, and liver. Secondary outcomes included mortality, duration of ICU stay, and duration of hospital stay. We used a phenol chloroform extraction of DNA from peripheral blood mononuclear cells using the venous blood sample. HSP70 genotyping was done using PCR-based methods. A PCR with restriction fragment length polymerase analysis was used to identify HSP-A1267G and HSP-A1L. Allele-specific PCRs were used to identify RS1008438 and the RS1043618 polymorphisms. Assuming the prevalence of the polymorphisms to be 55% and the prevalence of dysfunction in 3 or more organ systems to be 64%, taking a power of 80% and a type 1 error of 0.05 to detect an association with an odds ratio of 4 between the polymorphism and outcome, we calculated a sample size of 108. Categorical data were analyzed using a Pearson chi-square test, continuous data using a two-tailed independent t-test, SPSS was used for analysis, haplotype association analysis was done using HaploView. We recruited 108 patients, 48 male and 60 female, at a mean age of 40 years. The most common diagnosis was swine flu, scrub typhus, and urosepsis. Other diagnoses included dengue, community-acquired pneumonia, unidentified acute febrile illness, and others such as melioidosis. Dysfunction of the respiratory system was the most common and seen in about 84% of patients. 50 to 60% of patients had dysfunction of the renal, cardiovascular, liver, and hematological systems. 1 in 5 patients had dysfunction of the metabolic system, and 1 in 6 patients had CNS dysfunction. Now, this is a graph that shows the number of dysfunctional organ systems per individual. As you can see, 65% of the patients had dysfunction of 3 or more organ systems. The mortality was just shy of 40%. 17 patients did not require ventilation. 25 patients could be managed solely on non-invasive ventilation. 66 patients required invasive ventilation. The mean duration of ICU stay was 9 days, with most patients hospitalized for about 2 weeks. Now, this is a table that shows the distribution of the HSP70 genotypes. What I want you to look at is the heterozygous group. The majority of the patients are heterozygous and have at least one polymorphic allele. We noted that patients with one or more polymorphism had an increased risk for requiring invasive mechanical ventilation with a relative risk of 2. They also had an increased risk for developing hematologic dysfunction with a relative risk of 1.9. Hematologic dysfunction was also significantly associated with the HSP-A1267G polymorphism. It also showed a trend towards a significant association with the RS100-8438 polymorphism. The association between the polymorphisms and the number of organ dysfunctions per individual was not significant. Duration of ICU stay was significantly longer in those with the RS100-8438 polymorphism and those with the A allelotype of HSP-A1267G. We did a subgroup analysis in the largest group, which was the H1N1 patients. Again, the association with the number of organ dysfunctions was not significant. Liver dysfunction, however, had a significant association with the RS104-3618 polymorphism and showed a trend towards a significant association with HSP-A1L. Duration of ICU stay was significantly longer in people who had any one of these three polymorphisms, the HSP-A1267G, RS100-8438, and RS104-3618. Now, this is a table to point at the variation in the frequencies of these polymorphisms from population to population. In RS100-8438, the frequency of the A allele in this study was 40%. It's been noted to be as low as 8% in the sub-Saharan population and as high as 89% in the European population. Again, with the RS104-3618, the frequency of the polymorphic C allele was 57%. This is considerably higher than what has been noted in any other population. We were able to compare the other two polymorphisms with another study done in India on patients admitted with diabetic foot. Now, if you'll notice, in both the polymorphisms, in both HSP-A1267G and in HSP-A1L, the frequency of the polymorphic alleles is similar to the other study, being only 5% higher in this study. Now, this is an image that shows the haplotype association in A being in the entire cohort and image B being in the subgroup with H1N1. Now, what this shows is that the three polymorphisms, RS100-8438, RS104-3618, and the last one being HSP-A1267G, which are depicted by the red squares, show a significant linkage disequilibrium or non-random association, meaning that these three polymorphisms are likely to be present together in an individual. This indicates that these polymorphisms have probably been in our population for generations. In conclusion, single nucleotide polymorphisms of the HSP70 gene are frequent in patients with sepsis in our population. The HSP70 genotype was significantly associated with ICU stay as well as with hematologic and hepatic dysfunction. Although no association with mortality was noted, the study was not powered to detect such an association. The next step for us would be to conduct cytokine assays and compare the levels of the cytokines with the presence of the polymorphisms. Thank you.